Hello, Science 30, and welcome to our video on mitosis and meiosis. We are going to be comparing the two processes and discussing where each process occurs in our body. So as you can see from the little GIF that's going on, they are different. So let's discuss mitosis. So what is mitosis? Mitosis is essentially cell division. So in your notes, you want to add cell division. And it occurs in all of the cells of our body except for your sex cells. So no sperm or eggs go through mitosis. So we call those your somatic cells. All right. So anything that is somatic is known as a body cell. So that's where mitosis occurs. Mitosis occurs during the cell cycle. So the cell cycle has to occur where you have replication and then you have growth and then you're going to have division of the cell. And mitosis has to occur because if we didn't have mitosis occurring, we would never replace cells that we had. Our cells would die and then slowly but surely we would have no cells left and we would never be able to repair or grow our cells. So what we're going to do is we're going to draw the process of mitosis. I'm going to say stage 1, stage 2, stage 3. I may also bring up the names of those stages. You do not need to know the names of the stages. You just need to know the pictures and be able to identify them as stage 1, 2, or 3, and so forth. So let's start with stage 1. Stage 1, this one right here, is called interphase. In stage 1, you can see in the center here, we have the nucleus, and inside of there, we have replicated DNA. That's what all of this is. And this is just called the nucleolus. But inside this nucleus, we have all this DNA replicated, waiting to go through mitosis. So in stage 2 of mitosis, which we refer to as prophase, all of this DNA condenses down to make what we would call chromosomes. Now, chromosomes are what has genes on them. It's what tells you your hair color, your eye color, your height, those kinds of things. And in stage 2, what it's doing is it's prepping to get ready to divide. So we want all of the DNA condensed down because the nucleus has to disappear in order to make space for division, because you can't divide this in half. We're going to get rid of it so that we can just divide these in half. So in stage 3, all of the chromosomes line up on what we call the equatorial plate or the metaphase plate. Stage 3 is known as metaphase. So they're attached to what we call spindle fibers. That's these long blue structures here. And they attach to something called the centromere. Now, the centromere is at the center of the chromosome. And what's unique about chromosomes is even though they're in an X, the genetic material on this side is the same as the genetic material on this side. So when they split in two, they're getting identical copies to each side of the centromeres or centrioles, which are over here. So in stage 4, we call this anaphase. This is essentially when they break apart, they break up. Okay, so these two chromosomes here were in X form over here. They now get pulled to opposite poles. So they're getting pulled in opposite directions. This chromosome and this chromosome have the exact same genetic material. They have the exact same DNA. And it's the same for all of these as well. So as they get pulled apart, these are going to eventually make two new cells, and each cell is going to have the same DNA in it. So in stage 5, we have now what we would call telophase. And telophase is essentially creating two new cells. So you can see that this is splitting into two, and our chromosomes are going to go through elongation, meaning they stretch out again and slowly make DNA. Now, as telophase is occurring, another phase is occurring with it called cytokinesis. So as telophase is happening and we get this pinching off of these two cells, this is called the cleavage furrow. What ends up happening is it slowly divides until you get two brand new identical cells. And that is mitosis. This is happening in every cell in your body except the eggs and sperm cells. So I'm just going to go back and run through those one more time. So here's stage 1 as interphase. Please make sure you draw this diagram and label it as stage 1. Here is stage 2, which we call prophase, but again, you just need to call it stage 2, draw it, And so you can be able to identify it. Now, if you want to give some identifying features, you can just remind yourself these are chromosomes and these are centrioles. It's up to you. You just need to be able to identify the photo. Again, this is stage 3, or what we call, metaphase, stage 3. All of them are lined up at the plate. So please again, draw this. And then anaphase is stage 4. So label and draw this as stage 4. And last but not least, you can call these stage 5 and stage 6, which is telekinesis and cytokinesis. So within your notes, you should have all 6 of these stages drawn and just labeled as stage 1, 2, 3, 4, 5, and 6. So let's move into what we would call meiosis. Now, on the other side, you are going to draw the diagram of meiosis. Now, meiosis and mitosis are very, very, very similar. The only difference is meiosis is only for sex cells. So I said mitosis is for all your body cells. Meiosis is for sperm and egg. And the reason for that is because meiosis does 2 divisions instead of 1. In mitosis, we ended up with 2 identical cells. In meiosis, we're going to end up with 4 very different cells. The DNA in them are all a little bit different. And the reason for this is that is what we would call genetic recombination. When you get a little bit of different genetics in each egg and sperm so that when they come together, they make people that look different. You, as if you have siblings, you don't look exactly like your brother and sister. You're not identical. Okay? And that is because there's a little bit of genetic recombination occurring due to meiosis. So in stage 1 of meiosis, we see interphase again. This is stage 1. Now, meiosis is broken into 2 parts, meiosis 1 and meiosis 2. So this is stage 1 of part 1, essentially. And it goes through the same thing that it did in mitosis. This is called prophase, except prophase, or stage 2 in this case, or prophase 1, instead of just all the chromosomes just being randomly inside where the nucleus was, they now make pairs. And you can see these pairs right here. You can also see that some of the genetic material has been switched. And that's how we get genetic recombination. That's called crossing over. So in prophase 1, or stage 1-1, we'll call it, all right, the whole idea is to find your partner and make sure that you can switch some genetic materials with your partner. In stage 3, this is metaphase. In metaphase 1, or stage 3, part 1, you're seeing that they are also lining up at the metaphase plate. All right. So in stage 4, in anaphase, instead of 2 single chromosomes being split apart like they were in mitosis, full chromosomes are getting pulled to opposite poles. Now, this stage right here is what we call the reduction stage of meiosis, meaning you're getting half the genetic material going to each side. And I'll explain that a bit more once we get to discussing this. So once we get there, we're going through stage 5, which is telophase. And telophase again develops the cleavage furrow. We're going to redevelop our nucleus, and we're going to go through cytokinesis. So now I have 2 new cells that each have 2 chromosomes in it. If we go back to the original picture over here, you'll see that there was 4. Here was 4 in one cell. Now at the end of the very first division of meiosis, we have 2. And that is why it's called reduction. We're going from 4 to 2. And that is how our sex cells become 23 chromosomes instead of 46. Every cell in your body has 46 chromosomes in it, except for the egg and sperm, which only have 23. So that when an egg and a sperm fertilize, they end up with 46. Now, meiosis has 2 parts. So this was part 1 of the entire process. So make sure you label this as part 1, stage 1, 2, 3, 4, 5, 6. In stage 2, we now have 2 cells, each with half the DNA we started with, going through the exact same process we've already gone through. This is interphase / prophase. Here is metaphase, or stage 2 and stage 3. And here is stage 4, which is anaphase. All right, so we're just moving through the exact same phases again, except this time with 2 cells instead of 1. We're now in the telophase stage where we're going to start seeing some division of the 2 cells, and we're going to end up with 4 different cells. In this case, these are showing sperm cells and egg cells. So we'll pretend these are eggs and these are sperm, just pretend, okay? Now what you're going to see here is that each one of these is a little bit different than the other. In mitosis, when we split into 2 cells, the DNA was exactly the same. But in this case, in meiosis, you're going to notice that there is no 2 chromosomes in the inside of the nucleus that look the same. And that is the whole idea of why we have meiosis occurring, for genetic recombination and to reduce the number of chromosomes. So please make sure that when you go through this video that you are drawing every stage down and maybe using colors as you go through so that you can identify the difference between mitosis and meiosis. And that is essentially what your responsibility is to be able to pick out and identify each stage by looking at the picture. Perfect. So ladies and gentlemen, if you have any questions, please let me know and I will talk to you soon.